Okay guys, in our last video we learned about nucleic acids. We learned that we have two types of nucleic acid, the DNA and the RNA. We also learned that these nucleic acids are made up of small monomers called nucleotides, and we did a little bit of basic structure of the nucleotide. But in this video we're going to be more specific on the names of the structure of the nucleotide. For example, we'll go deep into the sugar and the nitrogenous bases like I explained. But the phosphate, remember, it doesn't change. But we're gonna focus on DNA. After then we will look at RNA in the next video. Okay, let's just jump in right into DNA right away. Okay, we're going to talk about the structure of the nucleotide of the DNA. Okay, we learned that the nucleotide has three parts: the phosphate, the sugar, and the nitrogenous base. Well, for DNA, the sugar is actually called deoxyribose sugar. The phosphate, they're the same, we just call it the phosphate. The nitrogenous base, we have two groups of nitrogenous bases. We have the purines and the pyrimidines. Okay, I don't really know how to pronounce the word, but it doesn't matter as long as I know how to write it, and it's not usually asked in exams. In these two groups, we have two nitrogenous bases in each group. Okay, let's start with the first one, the purines. In the purines, we have two nitrogenous bases, adenine and guanine. They each end with 9, so the suffix is 9. I don't know if I'm saying it right, but the suffix is 9. So I say the purines adenines. Then we have the pyrimidines. We have thymine and cytosine. These ones, okay, we only have 4 of them. So this means if you got your 9s with the purines, then the pyrimidines are the rest that are remaining. So it's going to be easier for you. So yeah, that's our 4 nitrogenous bases on DNA. Remember that it's gonna change on RNA, so this is just for DNA, okay? But they are similar, they only differ in small amounts. So yeah, okay, now that we know the basic structure of the nucleotide in DNA, we know the nitrogenous base, we know the sugar. I want us to now explore how does these nitrogenous bases form the double-stranded DNA. How does these nucleotides, okay, let's think about this. We have the cell, right? In the cell we have the nucleus. Inside the nucleus we have the chromatin network. When we unravel that chromatin network, we get chromosomes. As you guys can see, we get chromosomes. Then deep inside the chromatids, one of the chromatids, that's where we find our DNA. Now remember, DNA is double-stranded, right? So these strands are just a line, a connection of nucleotides from the top all the way to the bottom. Those are just nucleotides. Now it's really important for you to remember this. Now listen, we have 4 nitrogenous bases, right? Which are adenine, guanine, and cytosine and thymine. These are our 4 nitrogenous bases in DNA. So these nitrogenous bases bond in order to to create this double-stranded molecule. So, but these nitrogenous bases, they each have a specific base that they bond to. For example, adenine only bonds with thymine. So A only bonds with T. Adenine only bonds with thymine. Guanine only bonds with cytosine. So G only bonds with C. So each of these bases bond like repeating themselves over and over until like forming this large double-stranded molecule, guys. As you guys can see on the DNA structure picture over there, it shows that adenine, the green, only bonds with thymine, the blue. And you can look at the structure too. Guanine, the purple, only bonds with cytosine, the orange. So, and this silver stuff is just row of sugar, meaning deoxyribose sugar and phosphates. So yeah, I think that's it. That shows how the DNA strand is formed at a molecular level. Anyway, that's it for the video, guys. Please like and subscribe and follow for the next video, guys. Peace.